All right, in chapter 17, we're going to talk about viruses and bacteria. Protists are also included in this chapter at the end, but we are not going to cover those at all. It's just way too much information and we don't have time. Okay, so let's start with viruses. If we're talking about viruses, they're really like the rebels of biology because they don't play by the usual rules. In fact, they're not even included in the classification of living organisms. Why? Because they're non-cellular and they can't do anything on their own. They have to have a host. So that's why we call them obligate intracellular parasites. So whether viruses are alive or not is still a hot debate among scientists. Most consider viruses to be non-living. They can reproduce and evolve, which are characteristics of life, but they can't metabolize or respond to stimuli without hijacking a host cell. So every virus has two basic components. It's going to have an outer capsid, which is a protein shell made up of subunits that protect the virus, and then it has an inner core that contains its genetic material, either DNA or RNA, but never both. Compared to cellular organisms, the viral genome is really, really small, often just a handful of genes. So some viruses have extra features like spikes on their surface that are going to help them attach to the host cells, or some have an outer envelope that's made of pieces of the host's own cell membrane, which helps them sneak into new cells. So this is just showing you the influenza virus, kind of as a model to understand viral anatomy in more detail. So the flu virus has a spherical or oval shape and a lipid envelope derived from the host cell membrane. Studying the envelope are two types of spike proteins. We have hemagglutinin, which is abbreviated HA sometimes, and neuraminidase, or NA. So the hemagglutinin allows the virus to bind to host cells, while the neuraminidase helps the virus exit the host cell after replication. And these spikes are so important that we name the flu strains after them, like H1N1 or H3N2. Inside, the virus carries segmented RNA, which just means that its genome is split up into pieces. And it also has enzymes like RNA polymerase for replication. So this structure allows the flu virus to evolve rapidly, which is why we have to have a new flu shot every year. So viruses are highly specific to their host cells, so they can usually infect only one type of organism, and often just certain cells within that organism. So the infection starts with when a virus attaches to a receptor on the host cell. After entry, the virus is going to hijack the cell's machinery in order to make copies of itself. And this process differs depending on the type of virus it is. But for bacteriophages, which are viruses that infect bacteria, we talk about two main cycles of reproduction. We can either have the lytic cycle or we can have the lysogenic cycle. So what's the difference? In the lytic cycle, things happen really, really fast. The virus injects its DNA into the host bacterium, takes over immediately, and then uses the host to make viral components like its DNA and its proteins. So these parts assemble into new viruses, which then burst out of the cell via lysis, killing the cell in the process. So the cycle has 5 steps. It's going to have attachment, where the virus binds to the host. Then it's going to have penetration. That's the viral nucleic acid entering into the cell. Then it has biosynthesis, where viral parts are being made. And it has maturation, which is the assembly of new viruses. And then release, where the host cell bursts and viruses spread. In the lysogenic cycle, the virus takes a different approach. So after attachment and penetration, the viral DNA is going to integrate into the host chromosome and become what's known as a prophage. It's then going to lie dormant, kind of just hanging out inside of the host DNA and copying itself as the bacteria divides. So the host isn't harmed, at least not right away. But if the cell is triggered by stress, radiation, or chemicals, that prophage can reactivate and jump back into the lytic cycle, causing the host cell to produce viruses and to die. So this is just showing you what's going on during the lytic versus lysogenic cycles of a bacteriophage. So plant viruses operate a bit differently. They typically enter the plant through damaged tissues like wounds or insect bites. But once they're inside, they move from cell to cell through the plasmodesmata, which are just small channels between plant cells. So a famous example is the tobacco mosaic virus, or the TMV, which causes molted, discolored leaves and stunted growth. So here's the virus down here. So for a long time, the only way to control plant viruses was really just to destroy infected plants and control insect vectors, which are the organisms that spread the virus. But now, thanks to bioengineering, we can make plants resistant to viruses. So a great success story is the development of transgenic papaya plants in Hawaii, which are resistant to the papaya ring spot virus, or PRSV. So this is really a breakthrough that saved the local papaya industry. So animal viruses use the same basic infection processes as bacteriophages, but many have a membranous envelope that's made from the host's own plasma membrane, which helps them evade the immune system. So the herpesviruses are a good example of how sneaky viruses can be. So they infect the host and then they go latent, hiding in the nerve cells. Then when triggered by stress, they can reactivate and cause symptoms again. So that's why cold sores or genital herpes often comes back during stressful times. Another important class of animal viruses is the retroviruses, such as human immunodeficiency virus, or HIV, which causes AIDS. So retroviruses, nucleic acid is going to be RNA, but it's not just RNA. They use an enzyme called reverse transcriptase to make a DNA copy of their RNA genome. So that DNA then gets inserted into the host genome and then becomes a provirus that can remain hidden for years. So this is just walking you through how HIV reproduces step by step. So first, HIV is going to bind to specific receptors called CD4 receptors on T helper cells, which are cells that are really a key part of the immune system. The virus is then going to fuse with the cell membrane and release its RNA and its enzymes, including the reverse transcriptase, into the host cell. The reverse transcriptase enzyme is going to copy the viral RNA into a double-stranded DNA, which can then travel into the nucleus and integrate into the host DNA using another enzyme called integrase. Once it's integrated, the virus may become dormant, or it may activate and begin producing viral RNA and proteins. So these viral components are assembled into new HIV particles, which then bud off from the host cell, taking a piece of the cell membrane with it to form their own envelope. So over time, HIV is gradually destroying the host immune cells, leading to AIDS.